Hello friends, in this video we are going to do the classification of amino acids in which we'll be taking these 20 amino acids one by one, and we also have discovered 21st and 22nd amino acids. But see, if 21st amino acid is discovered, that is selenocysteine, you know. So can you tell me which is 5th amino acid, which is 6th, which is 10th? No, there's nothing like that. From 1 to 20, they're not given any numbers. And just because 21st and 22nd were discovered later, so they are given one number, right? But there is no amino acid like 6th or 12th like that. There's no such numbering, right? So now, in case of amino acids, we have to do the classification, and here we'll be doing. So in this, first we'll be taking is classification of amino acid. First is aliphatic amino acid category, in which we'll be doing these 5 amino acids: glycine, alanine, valine, leucine, and isoleucine. So now when we are going to do 20 amino acids one by one, for each amino acid, we have to learn 2 properties. One is its essentiality. It is essential, non-essential, or semi-essential. And the other property we have to see is its polarity. It is polar, non-polar, or less polar, more non-polar, all that thing we have to do. So 2 properties for each amino acid. If we do the cramming here, then in the end you will be having 40 properties, and then everything will be intermingled and you will forget all the things, right? So we will not cram this. The way I have for teaching here is that every time we'll be making structure of amino acid, and then from the structure I'll be teaching you how to take an idea, and you can easily tell it as polar, non-polar, or essential, non-essential, right? So let's get started with glycine first. So making the structure of glycine, carboxy on right, amino on left. I told you I will always make it like this. And then hydrogen here and the side chain. What is the side chain of glycine? You know, it is simple hydrogen atom here. Now tell me, this much structure we can make or not? This much structure in body can be made easily, right? We can make few amino acids. So this common structure we can make. But we have to see the side chain every time. We have to see the side chain, and just an idea you have to see we can make it or not make it in the body. So you know, this simple hydrogen addition in the body can be done. Therefore, I will say glycine is non-essential amino acid. We can make it in our body. It is non-essential. We do not require it in diet. And glycine is smallest, simplest amino acid. And there are no isomers for glycine because the central carbon is symmetric, because 2 valencies are occupied by same atom, that is hydrogen, right? Then let's write down the uses of glycine. Uses of glycine is for making heme. You know, the first step in heme synthesis is succinyl-CoA plus glycine amino acid is taken to make this complex compound heme. Then in glutathione, the 3 amino acids are gluta means glutamate, thia means cysteine. Thia is for sulfur-containing amino acid. Here we take cysteine because sulfhydryl group is present in cysteine. And when this 1 is written in the end, this 1 means the simplest, smallest amino acid, that is glycine. So mnemonic is in the name. Glutathione name is telling you it has glutamate, cysteine, and glycine, right? Another use of glycine is in the formation of creatine and also in the formation of serine amino acid. So I say glycine is non-essential, we can make it in body, and then glycine can make serine in the body, right? So can I say serine is also non-essential in body, it can be synthesized from this amino acid glycine. Now let's see this question, which amino acid is responsible for flexibility of proteins? When this question is asked, then answer is glycine. But let's understand how. You have seen the structure of glycine. You know, glycine has smallest side chain. It has smallest side chain. Because it has smallest side chain, therefore, this kind of amino acid, smallest amino acid, can fit in a small space. It can fit in a small space. When it can fit in a small space, and when this kind of amino acid is present in the structures of proteins, then it will create bends in the structure of protein. Bend means a sharp bend. You know, at the bend there's so less space, so only glycine can be fitted here. And ability to bend is known as flexibility. So glycine is responsible for flexibility of proteins, right? And therefore, because glycine can create bends in the structure of protein, so it is found in beta turn or beta bend. Beta turn or beta bend. What is this? Let's say there's a polypeptide chain in this direction, another polypeptide chain is going in this direction. Now we have to join these two polypeptide chains, and this kind of structure, beta bend or beta turn, is required here, and only two amino acids are responsible in beta bend. That is, glycine can be found here, and another is proline can be found in beta turn or beta bend, right? And this amino acid glycine is never found in alpha helix. Because of same reason, it will create bend in the structure. Alpha helix is a very symmetrical helical structure. There is no sharp bend in the structure of alpha helix. So glycine is never found in alpha helix. Now let's see how glycine is synthesized and how glycine can make serine in the body. So carbon dioxide and ammonia, they will combine to make glycine. And here the enzyme responsible is glycine synthase. And for this reaction, vitamin B6 and vitamin B9 both are required. And this reaction in the opposite direction is known as glycine cleavage system. That is, glycine will be cleaved into carbon dioxide and ammonia again, known as glycine cleavage system, right? And now glycine can be converted to serine. And here the enzyme required is serine hydroxymethyl transferase, because hydroxymethyl group is getting transferred in this reaction. And again in this reaction also vitamin B6 and B9 both are required, and this reaction can go in opposite direction also. This is a reversible reaction requiring the same enzyme. And after that serine can be converted to pyruvate, right? Now glycine can also be converted to glyoxylate. Glyoxylate has two fates in the body. One is formation of formate, and this formate can enter one carbon pool of the body. On the other hand, glyoxylate will be converted to oxalate, which will be excreted in urine. And also glyoxylate can be converted back to glycine, and this is done by enzyme. One glycine transaminase enzyme is required here, right? Now see this detail that glyoxylate can be converted to glycine, or it can be converted to formate, or it can be converted to oxalate. Now let's see what happens in primary hyperoxaluria. If this transaminase, glycine transaminase, is not working, plus also glyoxylate is not getting converted to formate, then this excess of glyoxylate will be converted to oxalate, and this is known as primary hyperoxaluria. Primary hyperoxaluria. And in this patient will be having oxalate stones in urine because of excess of oxalate coming in urine. And in this disease, primary hyperoxaluria, there should be restriction of oxalate rich foods to these patients. For example, green leafy foods and beetroot also, and tea also should be restricted in these patients, right? And on the other hand, there is also secondary hyperoxaluria. Secondary hyperoxaluria means it is occurring because of some other thing, not because of primarily oxalate increase, but because of some other thing. For example, excess vitamin C can cause secondary hyperoxaluria. B6 deficiency can also cause ethylene glycol poisoning, and methoxyflurane, they all can cause secondary hyperoxaluria. Now coming to next amino acid, alanine. Let's make the structure. Alanine structure will be carboxy here, amino here, hydrogen here, and the side chain, you know, is a simple methyl group here. Now the simple methyl group also can be added in body. We can add this methyl group in the body. So again, alanine is also non-essential. And see here, alanine is a 3-carbon compound. Any 3-carbon compound is a good substrate for gluconeogenesis. Many amino acids can form glucose, but this alanine is most glucogenic amino acid, and also it is a 3-carbon amino acid. Now let's do Cahill cycle or glucose alanine cycle. Let's say this is muscle and this is fasting muscle. This is fasting muscle. This is liver here, and in the center here I'm making blood. So in fasting muscles, what will happen? Glucose will be converted to pyruvate, and then pyruvate will be forming alanine. Alanine can come out in blood. From blood, it is taken up by liver. In liver, alanine will be again converted to pyruvate and then finally forming glucose here. This glucose is supplied to that fasting muscle again. So this is a cycle formed known as glucose-alanine cycle, also known as Cahill cycle. And you can see here that mainly amino acid alanine is forming glucose in liver by gluconeogenesis pathway. So alanine is regarded as most glucogenic amino acid. So we're done with two amino acids in aliphatic category, that is glycine and alanine. Both are non-essential, not required in diet. But the other three I'm going to take together. Other three are leucine, valine, isoleucine. Valine, leucine, isoleucine. All three in their structure, in their side chain, they have some branch in the side chain. So they are called branch chain amino acids, and branching is a difficult thing in the body. Just an idea I'm giving you to learn. Branching is a difficult thing in the body. So all these branch chain amino acids are essential. We cannot make them in body, so we have to take them in diet. Now there's one disease for these branch chain amino acids, that is maple syrup urine disease. Maple syrup urine disease. This disease is a defect in the catabolism of these three amino acids, branch chain amino acids, valine, leucine, isoleucine. It is a defect in the catabolism. During their catabolism, there is one step of oxidative decarboxylation which is occurring. In a normal person, this oxidative decarboxylation step is occurring. But in MSUD patients, this oxidative decarboxylation step is not occurring. And the name of this enzyme is alpha keto acid dehydrogenase. You know that the name of enzyme of oxidative decarboxylation is dehydrogenase always, right? When I have written alpha keto acid here, so you can get this thing that alpha keto acid is the substrate of this enzyme, because name of enzyme tells you the name of substrate, right? So the name of enzyme is dehydrogenase, or also they can call it alpha keto acid decarboxylase. Both names are used for this enzyme. So whatever is given in the option, you can mark, both will be correct. Alpha keto acid dehydrogenase or alpha keto acid decarboxylase, right? Now in the clinical feature, these patients will be having, what is the odor of urine of these patients? Urine odor is maple syrup like, that is a sweet kind of smell coming from the urine. Typically we use the word burnt sugar like odor coming from the urine. And one clinical feature is given in the name of the enzyme. And you know the name of substrate is alpha keto acid. Alpha keto acid. So because alpha keto acids will be accumulated because this enzyme is not working, so these patients will be having ketosis. And these patients of MSUD also have mental retardation. Also, these patients have abnormal muscle tone because brain is affected. So these patients have abnormal muscle tone. And this disease, maple syrup urine disease, can be lethal also. That is, if it is not treated, then coma and death can occur. And these patients have high mortality rate. Death can occur, and in fact they have high mortality rate, right? Now there's one more disease in this branch chain amino acid category. Here only for leucine. The first one, maple syrup urine disease, we have done, which is for all the three branch chain amino acids. But this one, isovaleric aciduria or acidemia, is a defect only for the catabolism of leucine, and the enzyme deficient is again some dehydrogenase. It is isovaleryl CoA dehydrogenase. And these patients have cheesy odor of urine, right? So we're done with the aliphatic amino acid category. First two, glycine, alanine, were non-essential, and the other three, valine, leucine, isoleucine, are essential. So now we have to learn the polarity or non-polarity of aliphatic amino acids, and it is very easy for all the aliphatic. All the aliphatic amino acids are non-polar, in which isoleucine is most non-polar, followed by valine, and least non-polar is glycine. Least non-polar is glycine, most non-polar is isoleucine, But there is a controversy for the polarity of glycine. Some books say it is polar, some books say it is non-polar. Because it is least non-polar, so sometimes it is considered polar also. Once you know it is a controversy, you can solve your questions. For example, I'll give you example of 2 questions. So as there is a controversy for the polarity of glycine, so solve these 2 questions. Question number 1, which is polar. Options given are glycine, alanine, valine. Now all are non-polar. So all are aliphatic, all are non-polar. So which one will you mark? You will mark glycine because glycine is least non-polar, right? Now see the second question, which is polar. Now options given are glycine, alanine and aspartate. You know aspartate is negatively charged, so very clear aspartate is polar here. So now you will mark aspartate, now you will not mark glycine. So according to the question you will solve. Now next category I'm going to take is aromatic amino acid category, in which we'll be doing these 4: phenylalanine, tyrosine, tryptophan and histidine. So first 2 I'll be taking together, phenylalanine and tyrosine. Why we take it together? Because phenylalanine gets converted to tyrosine in body with the help of enzyme phenylalanine hydroxylase, right? So first we'll be making the structure of phenylalanine. Phenylalanine, the common structure, first I will make carboxy on right, amino on left, hydrogen here and the side chain here. Now for making the side chain, see the name is telling you the structure. The name says alanine. So first I will make alanine, right? And then add phenyl ring to this alanine. So here you add phenyl ring and that is the structure of phenylalanine. I have not completed the valency here. I'm not concerned with the valency here. There's no need for doing the valency in detail. You just need an idea of the structure, right? Now see the idea. Idea is this side chain, a big side chain present in phenylalanine. So you tell me an idea. Can it be made in body? This big complex side chain cannot be made in body. So we will say phenylalanine is essential. We require it in diet. And these kind of rings, benzene ring, phenyl ring, they are always non-polar rings. So we will say phenylalanine is non-polar, right? Now phenylalanine gets converted to tyrosine in body, with the help of enzyme hydroxylase. And whenever hydroxylation reactions are occurring, in the substrate only one O is added. But the group which you can see which is added is OH group which is added. That's why these are known as hydroxylases, right? So in this structure of phenylalanine, if I make OH here, because just one change is there. If I make OH here, then it will be the structure of tyrosine. Now tell me, it is essential or non-essential? We will say it is non-essential because we can make this tyrosine in body from phenylalanine, right? And because OH is present, that's why it is polar. But again, there is a controversy for the polarity of tyrosine. Some books say it is polar, some books say it is non-polar. So according to the question, you will solve, right? So we're done with these 2 amino acids, phenylalanine and tyrosine, which we have taken together. And now coming to the third aromatic amino acid, that is tryptophan. Let's see the structure of tryptophan. Tryptophan structure is very complex. First I made the common structure, right? Now the structure is complex, but we don't need the exact structure, right? We just need an idea of the structure. And the idea is this, that there are 2 rings in the side chain. Just an idea, there are 2 rings in the side chain. These 2 rings are known as indole ring or indole nucleus. So now you tell me, essential or non-essential? Because there are 2 big rings in the side chain. So we cannot make this kind of amino acid in body. So it is essential. Then these rings are always non-polar. So we will say that tryptophan is non-polar amino acid. And this kind of big side chain having 2 rings, this cannot be accommodated in alpha helix because alpha helix has very less space in the helical structure and inside very less space. So this is never found in alpha helix. So tryptophan is essential, non-polar and not found in alpha helix, right? Now see the uses of tryptophan. One use is that tryptophan will form serotonin and then it can form melatonin. So melanin is coming from tyrosine, but melatonin is coming from tryptophan. One more very important use of tryptophan is that tryptophan can form a vitamin, that is niacin, vitamin B3 is formed by tryptophan, and during the formation of niacin, this is a big pathway for which we are not doing the detail, but in this pathway vitamin B2 and B6 are also required. So can I say that vitamin B2 and B6 deficiency will also lead to vitamin B3 deficiency, right? Now focus here that this is a vitamin. Vitamins usually cannot be formed in body, right? We have to take vitamins in diet. But this vitamin, vitamin B3, that is niacin, is formed in body. So it is known as atypical vitamin. We have only two atypical vitamins, vitamin B3 and vitamin D, because only these two vitamins can be formed by our enzymatic systems during our metabolism, right? When I talk of atypical vitamins, then do not consider those vitamins which are synthesized by intestinal flora, right? Do not consider them. Consider those vitamins which are synthesized by human body enzymes, not by bacterial enzymes, okay? Now coming to catabolism of this amino acid, tryptophan, I'm not going to take the detail of complex catabolic pathway, but only one clinical thing which is required, that we'll be doing here. So tryptophan catabolism, during catabolism, there is one intermediate formed, that is 3-hydroxykinurenin. After that, next intermediate is formed, right? But in this reaction, kinurenin hydroxylase enzyme is required, which requires vitamin B6, that is pyridoxal phosphate, PLP is required. So in PLP, in B6 deficiency, in vitamin B6 deficiency, this 3-hydroxykinurenin will increase, and excess of this will be taken for another pathway which will form xanthuranic acid, and this xanthuranic acid is there in excess in body, which will be coming in urine. So we can use it for diagnosis that in B6 deficiency, xanthuranic acid is coming in urine, right? Now there's one disease for tryptophan, that is Hartnupp's disease. Hartnupp's disease is autosomal recessive. There is failure to reabsorb tryptophan from kidneys. So tryptophan will be coming in urine, and the clinical feature of these patients will be amino aciduria, mainly tryptophan coming in urine. Also these patients have pellagra-like symptoms. Why pellagra-like symptoms? Because you know that tryptophan is responsible for the synthesis of niacin in body, and now niacin deficiency will lead to pellagra. Also one point in carcinoid syndrome, all the tryptophan is used for the formation of serotonin in these patients, so that there is almost no tryptophan left for niacin formation. So these patients will also be having pellagra-like symptoms. So now we are done with tryptophan. So let's do the fourth aromatic amino acid, that is histidine. In the side chain of histidine, there is one pentacyclic ring present with 2 nitrogens in the ring, and the name of this ring is imidazole ring. Imidazole ring. In tryptophan, that was indole ring or indole nucleus. In histidine, it is imidazole ring. And see, the valency of nitrogen is 2. Now see the structure of this ring. Valency of nitrogen is 3. 2 valencies are already occupied in the ring, so that we have space for 1 hydrogen. And here also it is NH. So I told you the basic point that whenever a compound has amino group, that compound will be ionized to give positive charge, so that compound will be polar, right? But whenever amino group is present in the ring, it is not NH2, it is NH. NH will also be ionized, but it will be not fully ionized. It will be giving positive charge, but less positive charge, right? So we will say that histidine is polar, but it is less polar. Also, histidine is essential, but it is semi-essential, and histidine has maximum buffering capacity. Histidine has maximum buffering capacity. Okay, so these 3 things about histidine you have to learn. So we're done with aromatic and aliphatic amino acid category. Next two category of amino acids, that is basic and acidic amino acids, I'm going to take together. First is basic amino acid category, and second is acidic amino acid category. All the basic amino acids are positively charged, all the acidic amino acids are negatively charged. So I can say both these categories are having polar amino acids because they have charge. Then basic amino acids all are essential, and acidic amino acids all are non-essential, right? Now let's see the example. Example of basic amino acid is arginine, lysine, and can I take histidine also in this category? Histidine, because histidine also has positive charge. I told you it has positive charge but less positive charge, right? But it has positive charge. So arginine, lysine, histidine. This way, when I have already written in the decreasing order of polarity. Arginine is most polar, then lysine and then histidine. Arginine has 3 positive charges, lysine has 2 positive charges, and histidine has 1 positive charge. Arginine is semi-essential. Histidine is also semi-essential, but this lysine is essential. So we can say that all amino acids in this category are essential. 2 are semi-essential, but still we can say that all are essential, right? Then acidic amino acid category examples are aspartate, asparagine, glutamate, and glutamine. All these amino acids can be synthesized in body from TCA intermediates. I told you in TCA cycle, oxaloacetate, OAA is the short form of oxaloacetate which I write. Oxaloacetate can form aspartate, can form asparagine. Alpha ketoglutarate intermediate of TCA cycle can form glutamate, which can form glutamine. So all these 4 amino acids can be synthesized in body. So we will say that all are non-essential. They can be synthesized in body, right? Okay, now out of all amino acids, if they ask you which is most polar, then arginine because it has 3 positive charges, and most non-polar is isoleucine, which is aliphatic amino acid. Now solve this question. Which amino acid will move fastest towards cathode? Which will move fastest towards cathode? See, valine. Valine is non-polar. Aspartate is having negative charge. Histidine has 1 positive charge. Arginine has 3 positive charges. So what is the answer? You know cathode is negatively charged, so answer will be arginine. Be careful, cation is positive, anion is negative, but cathode is negative, anode is positive, right? Be careful with this thing. Now next category which I am going to take is OH containing amino acid category. Example is serine, threonine, and can I take tyrosine also in this category? Tyrosine having OH group, it is also belonging to aromatic amino acid category, right? Now about threonine, one special thing I have told you that threonine has 2 asymmetric carbon. So I told you it is essential. It is very difficult to make some amino acid having 2 asymmetric carbons in the body. So threonine is essential. But serine is non-essential because I told you it can be synthesized from glycine, and tyrosine is also non-essential because it can be synthesized from phenylalanine, right? Now can I say all the OH containing amino acids are polar? OH group means polar because it can make hydrogen bonds, right? Now see these 3 questions, because whenever these 3 questions are asked, your answer is OH containing amino acid. See the first question, which amino acid has maximum tendency to bind phosphate? You know the basic point, OH has tendency for phosphate or phosphate has tendency for OH. So here answer will be OH containing amino acid. Now see the second question, which amino acid is the site for covalent modification? Covalent modification again means phosphorylation and dephosphorylation. So again your answer will be some OH containing amino acid, whichever is given in the option. Next, question number 3, which amino acid is responsible for O glycosidic bond? O means we have to take oxygen, so that oxygen will be taken from OH containing amino acid, right? There are 2 kind of glycosidic bonds, O and N glycosidic bond. If they ask you N glycosidic bond, which amino acid is responsible, then you will say it is asparagine because asparagine has CONH amide bond and this nitrogen can be taken from this amide bond. Now, I was saying that whenever these 3 questions are asked, your answer is OH containing amino acid. But if by chance in a question, all the OH containing amino acids are given in the options, then, and you have to select one out of them, then out of these 3, the best answer will be serine, which you can mark, right? Now coming to sulfur containing amino acids. sulfur containing amino acids are two, cysteine and methionine. Cysteine, methionine. Cysteine is having sulfhydryl group, but methionine is not having sulfhydryl group. In methionine, sulfur is attached to two carbons with a strong bond, and only in cysteine we have sulfhydryl group. Because of the sulfhydryl group, cysteine is polar, but methionine is not having sulfhydryl group, so it is non-polar. Now methionine, we are obtaining methionine from diet, so it is essential. It is very difficult to attach sulfur with two carbons. So this cannot be done in body, so we require methionine from outside. But in body, methionine can be converted to cysteine. Therefore, I will say cysteine is non-essential. So can I say cysteine becomes essential only if methionine is not available in diet? And I can also say tyrosine becomes essential only if phenylalanine is not available in diet, right? Then the last amino acid here is amino acid, IMINO, amino acid. Example is proline. Only one example here, proline. When we say amino, IMINO, means amino group is not free here. Because amino group is not free, therefore this kind of amino acid will disrupt the secondary structures of proteins. So it is not found in alpha helix. Proline never found in alpha helix. Proline is found in beta turns and beta bends. I already told you. Glycine is also found in beta turns. And proline is non-polar, non-essential. Non-polar, non-essential. So with this, we are done with all 20 amino acids. Now let's do 21st and 22nd amino acid also. 21st is selenocysteine given by codon UGA. 22nd is pyrrolysine given by codon UAG. You know that both these are stop codons. Stop codons means they do not code for any amino acid. General line, stop codon means they do not code for any amino acid. But now you can see this like an exception that stop codons can give rise to two amino acids, the 21st and 22nd. By some modification of stop codon, right? And this modification is co-translational modification, not post-translational modification. For example, you know in collagen fibrous protein, post-translational modification will convert proline and lysine to hydroxyproline and hydroxylysine. The post-translational modification is known as hydroxylation here in collagen, right? But in case of selenocysteine pyrrolysine, stop codon modification is occurring, but it is co-translational modification. That is, during the translation, this modification will occur, not after the translation, right? Now, there are few enzymes which require selenocysteine, the 21st amino acid at the active site. Those enzymes are known as selenoproteins. Mainly, they are reductases and peroxidases. Reductases and peroxidases. Example, you should note it down. It is very important. Glutathione peroxidase, thioredoxin reductase, and iodothyronine deiodinase, right? Now, precursor amino acid for selenocysteine is not cysteine. The precursor amino acid is serine. See this, I told you glycine will form serine by this enzyme, serine hydroxymethyl transferase. Now, in serine, if you add selenium, it will be selenocysteine, the 21st amino acid. And also, serine can be converted to cysteine by adding it with homocysteine. This homocysteine is derived from methionine, and homocysteine will be converted to cysteine, and here vitamin B6 is required. So can I say for the synthesis of cysteine amino acid from methionine, we require two amino acids, serine, methionine, and we require one vitamin, that is vitamin B6, right? So with this, we are done with all the amino acids. Now, individually, I have told you which is polar, which is non-polar. Now, together all I am combining, and I am taking this polar amino acid thing. Polar amino acid can be divided into two types, charged and uncharged. Charged polar are because of their charges. Like acidic amino acids are negatively charged, glutamate, aspartate. Basic amino acids are positively charged, lysine, arginine, histidine, right? But there are few amino acids which are polar, but they are not polar because of their charges. They are polar because of some other thing. Let's see the examples. First is OH containing amino acids are polar because of OH group. Then cysteine is polar because of its sulfhydryl group. Then asparagine, glutamine are polar because of their amide group, that is CONH group, right? Now let's also see derived amino acids. Derived amino acids are those amino acids which are not coming from codones. Let's see the example. Hydroxyproline, hydroxylysine, which are found in collagen, and they are post-translational modifications of proline and lysine to make hydroxyproline and hydroxylysine. It is done by hydroxylation, right? Other two, ornithine, citrulline, are involved in urea cycle. Homocysteine is involved in methionine metabolism, and homocysteine can further be converted to cysteine, right? Now, so we can see these two derived amino acids are seen in proteins, but these three derived amino acids are not present in proteins, right? This is it with this topic. Thanks for watching.